Okay, so this week we're going to finish up eukaryotic diversity by looking over the last two lineages that we did not cover last time. So specifically, we're going to look at Strontimanipula and the lineage Plantae, which includes the kingdom Plantae. One thing that you might have noticed as we've been going through each one of these different groups of organisms so far is that they're becoming more and more complex. And we're getting to that point where we really need to talk about the different types of reproduction. So since everything that we're talking about today and the previous class is under the domain eukarya, these are all eukaryotes, meaning they can have diploid cells. And by diploid, we mean all the chromosomes come in pairs. All right, so that's important to keep in mind as we go through these different types of reproduction. All right, so two major types of reproduction in eukaryotes is asexual and sexual reproduction. So with asexual reproduction, cells are going to divide and multiply through mitosis. And if you might remember, with a diploid cell under mitosis, you're going to end up with two diploid daughter cells. So looking at this picture here, we have two different forms of asexual reproduction. Both of these are eukaryotic cells, meaning their nuclei are diploid. So they have two copies of every gene in this parent cell for both of them. And through asexual reproduction, you're going to duplicate those two copies of a gene. So you technically have four copies of that gene before the cell splits, and then two of the copies go one direction while two of the copies go the other direction, and you're back to two diploid daughter cells. Because that's happening in both of these cells right here, since we're talking about eukaryotes, and most eukaryotes are diploid. However, there's different types of asexual reproduction, and here's just two major ones. One, we could have binary fission, or we can have something called budding. The binary fission is pretty straightforward. What is happening here is that the nuclei are duplicating, and the cells are going to first split their nuclei. So you have a diploid nuclei at one end and a diploid nuclei at the other end, and then the cell itself is going to split. So you have two identical cells at the end. However, with budding, it's a little different. So with budding, you have a daughter cell or a bud that kind of breaks off the side of the parent cell. So the first thing that's going to happen is the nuclei is going to duplicate all of its chromosomes. So you actually have four copies of any gene right before budding occurs. Then two copies go into the bud and two copies stay behind in the parent, and you're left with, again, a diploid and a diploid cell. Now, the major difference here is that the new cell is actually much smaller than the parent cell and needs to grow afterwards. Okay, so asexual reproduction, we have mitosis occurring. And asexual reproduction is really great if you don't need genetic variation. If you are well adapted to the environment and you just need to multiply as fast as possible, asexual reproduction usually is the way to go. However, not all environments are static. So environments are constantly changing and you need your genes to change with them. All right, and that is the benefit of sexual reproduction. So there's three major types of sexual reproduction that we're going to talk about here, and it has to do with the gametes that are being made. So by gametes, we mean sperm and egg, or the equivalent of a sperm and egg. So the three major types of sexual reproduction we're going to talk about is isogamy, anisogamy, and oogamy. So with isogamy, this occurs when your gametes, your male and female gametes, or different sexed gametes, look identical. So usually they're both motile, as you can see here, so they could swim around. They might be flagellated. And then they're going to swim around until they meet and then fertilize each other, creating a new organism. So one thing to keep in mind is that these gametes are made through meiosis, not mitosis. So even though the parent cell that these gametes came from were diploid, each of these cells that were made after are going to be haploid. These are your gametes, haploid gametes. So in isogamy, both the gametes are able to move, and they're going to find each other and fertilize each other. And when they fertilize, this haploid gamete and this haploid gamete fuse, making a diploid zygote. All right, next we have anisogamy. And anisogamy is very similar to isogamy, where both the gametes are mobile, meaning they both can move around, so they're swimming towards each other. However, you could tell the male from the female in anisogamy. You could not do that in isogamy. You would not know which one's the male and which one's the female until one of them donated their DNA. Here, you will know immediately who's going to donate their DNA because the male will always donate the DNA. And here, the males are usually smaller than the female's gamete. However, in anisogamy, they both can move. And then finally, we have something called oogamy, and oo means egg in Latin. And in oogamy, we have an actual egg. We have this stationary female gamete that doesn't move. And then we have this mobile sperm that is going to swim and try to find the egg to fertilize it. All right, so these are just three major types of sexual reproductions, and different organisms use different ones of these. So we're going to see a couple examples of these as we go through our eukaryotic diversity. All right, the next thing we need to talk about is types of life cycles. All right, different organisms will spend a certain amount of time either in a diploid or haploid stage. And so there's key differences between these life cycles. I'm going to start down here because this is probably the one most familiar to you. This is a diplontic life cycle, meaning the adult is a 2N stage, and then we have a very brief haploid stage of the life cycle where you're just going to meet gametes. And when these gametes are made, they fertilize each other and you make another 2N adult zygote. This should be familiar to you because this is what we do as humans. We spend our whole life as 2N. All our cells are 2N, meaning they're diploid. We have pairs of chromosomes. That's all that means. And then we make our gametes through meiosis. So we're going to have haploid gametes, which is sperm and egg. And then the sperm and egg will fertilize each other, making another diploid adult. Not all organisms do that, though. All right, some can have a haplontic life cycle, where they actually spend most of their life in a 1N stage, and then have a very, very brief period where they're 2N. So you're going to see this a lot in fungi, and you'll probably see this again when we come back later on the semester to fungi. But fungi spend most of their time in an N stage. So 1N meaning haploid. So you have haploid cells. They can develop structures that are going to be haploid. Eventually, they're going to release gametes that are haploid. And those gametes will fertilize to create a diploid zygote. And this usually will undergo meiosis immediately after to create new haploid cells. All right, and the last one that we're going to talk about, which we're going to see a lot of in this lecture, is alternation of generations. This is something we see a lot of in plants. So plants actually have two distinct life stages. They have a sporophyte stage and a gametophyte stage with distinct ploidy. So the sporophyte stage is their diploid stage, while their gametophyte stage is their haploid stage. So we call this alternation generations because this gametophyte might be a certain plant that grows somewhere. It will release gametes that'll fertilize to create a sporophyte, which is a different adult stage that will grow somewhere else, which will eventually undergo meiosis to create spores that will be released as gametes somewhere else again. All right, and so this is why we call it alternation generations, because the first generation might be haploid, while the second generation is diploid, while the third generation is haploid, and back and forth throughout this life cycle. And this actually brings up something I do want to distinguish, which you're going to see, and that's the difference between gametes and spores. The major difference between spores and gametes is that spores do not fertilize each other. That spore, that single cell, is going to divide and grow up to be this structure or this adult stage. However, gametes, even though they're single cells, need to fertilize each other. When they fertilize each other, they make a zygote, which will then grow to be the sporophyte. So that's the major difference between spores and gametes. Spores do not fertilize each other. They just start dividing, while gametes do fertilize each other. Okay, just showing you this alternation of generations again in plants, and also some of the protists under the lineage Plantae will do this as well. So let's start here. We'll have some adult sporophyte. If you remember, sporophyte is 2N diploid. They will have a structure called sporangium that's going to make spores. These spores are going to be made through meiosis. So if this diploid structure, this sporangium, is making spores through meiosis, what ploidy do you think these spores are going to be? Well, if you remember from GB1, through meiosis, the product is haploid cells. So you start with a diploid cell, goes through meiosis, and you end up with haploid cells. So these spores will be 1N, so haploid. Again, spores don't fertilize each other. Each one of these individual spores will be flung off into the environment. Well, they'll grow into a gametophyte, which is the next adult stage. So now we have this next adult plant that is actually haploid. So you'll see a plant that is haploid, meaning they're 1N. All right, this adult plant is going to make two structures called archegonium and antheridium. These are your sexual organs. With archegonium, it's going to make eggs, while the antheridium is going to make sperm. Now, this gametophyte is 1N. Just like we said, this archegonium and antheridium is 1N, so each of these are haploid. And then they make haploid gametes. So these gametes are actually made through mitosis. And if you remember with mitosis from GB1, you always end up with the same ploidy that you started off with. So if the antheridium is before mitosis, which is one end, then we have mitosis occur and we make a sperm through mitosis. This sperm is still going to be one end. You ended with what you started with. So the sperm is going to go and fertilize an egg somewhere. And when you fertilize, you have this haploid cell meeting this haploid cell, creating a diploid cell. And that first diploid cell that's made through fertilization is called a zygote. So that one cell, that zygote, is going to start to divide through mitosis. And again, mitosis, you end with what you started with. So if this diploid zygote's undergoing mitosis, we're going to have a bunch of cells that are diploid after mitosis to develop an embryo, which is a young plant. This embryo will then eventually grow up to be the other adult stage, that sporophyte again, which is also 2N. Okay, so now we're going to get into the diversity that we missed last time. So specifically, we're looking at the lineages Plantae and Stramonipola. We're going to spend a little bit more time on Plantae since it was so important to giving rise to plants, to land plants. So we're going to look at a whole bunch of different examples of organisms that fall under that lineage. And then we're going to talk about Stramonipola, and we have a few examples for that one. Also important species. All right, under the lineage Plantae, we're going to look at these groups. We're going to look at Rhodophyta, which is a phylum for red algae. We're going to look at Chlorophyta, which is the phylum for green algae. And then we're going to look at the kingdom Plantae. So green algae were believed to give rise to land plants. They have a lot of things in common with land plants, and especially at the molecular level. A lot of the DNA is similar. So it's pretty confident. So we're pretty confident that plants, the first plants, evolved from a green algae. All right, then we're going to look at the diversity of plants. So we're going to look at non-tracheophytes, which is a clade of plants that lack vascular tissue. Then we're going to look at tracheophytes, which is a clade of plants that do have vascular tissue. Underneath these tracheophytes, we're going to look at two different types. So we're going to look at seedless tracheophytes, which are plants that have vascular tissue but don't make seeds. And then we're going to look at two types of seeded tracheophytes, specifically gymnosperms and angiosperms. Now, under these two clades that make seeds, gymnosperms will not make a fruit, while angiosperms will make a fruit. Then we talk about Stramonipola. There's 3 phylums that we're going to cover. We're going to talk about Bacillariophyta, which is your diatoms. Then we're going to look at Mycota, which is your water molds, which is actually a type of protist, not a fungi. And then we're going to look at Phaeophyta, which is your brown algae. All right, underneath the lineage Plantae, we're going to start off with the phylum Rhodophyta, which is your red algae. Now, something to keep in mind as we go through the lineage Plantae, they all have something in common because they're all related to each other. They all came from the same common ancestor. And one of the characteristics that all of these organisms are going to have in common is they're all going to be photosynthetic, and they're going to have cell walls that contain cellulose, just like plants do. Plants contain cellulose. All right, so this is one of those pieces of evidence that they're all related. All right, looking at Rhodophyta, your red algae, they have a couple of unique characteristics. So when we're talking about photosynthetic organisms, they're all going to have a special photopigment called chlorophyll A. Chlorophyll A does most of the work for photosynthesis. However, organisms that are photosynthetic can be different in how they have accessory pigments, or what accessory pigments they have. So one unique pigment that rhodophytes have is something called phycoerythrin. This is a photopigment we saw in cyanobacteria. Unlike a lot of the other organisms under the lineage Plantae, Rhodophyta has a lot of phycoerythrin. It is their dominant secondary pigment. And you might remember when we were talking about phycoerythrin, erythri, means red in Latin. All right, so this pigment gives off a red color. Photopigments that look red are actually very good at absorbing colors that are in the blue-green spectrum. All right, they look red because they're reflecting that color red, but they're really good at absorbing blue-green or taking energy from blue-green. And that gives a hint to where we find these red algae. We actually find them in deep ocean or deeper water. And that's because the wavelengths of light that can penetrate the deepest in the ocean is blue and green. And that's why the ocean looks blue. And that's because that wavelength for that color can travel the furthest. So this gives these organisms a special advantage in deep water. So they will outcompete anything that is in deep ocean or deeper water where blue light is really the only light available to undergo photosynthesis. So these organisms also have a couple of different chemicals in their cell walls that we use commercially. So this is where we get a lot of the material to make agar and carrageenan. Now, carrageenan is something that we see a lot in toothpaste. It kind of gives that thickness to our toothpaste that we use. And if anybody's done anything with bacteria before, you should be familiar with what agar is, like an agar plate. So it's this really gel-like substance that we use a lot. So these are commercially beneficial organisms. Okay, so one unique characteristic that red algae has is a triphasic life cycle. And this is very rare. This is a rare thing to see, this triphasic life cycle. So red algae do display this. So that's what this picture right here is depicting. So we can go through these steps. Now, if you remember from your alternation of generations, let's start here at the gametophyte. With alternation of generation, you might have remembered that gametophytes are haploid. So they'll have a 1N adult stage. This adult gametophyte is going to make gametes. So it's going to make a sperm and egg, and the sperm will then fertilize an egg, and when that happens, you have a zygote, and that zygote's going to grow up to be the carposporophyte, which is the next adult stage. So now you have this 2N adult stage. This carposporophyte is going to make something called carpospores, and if you remember, spores don't fertilize, and it makes these carpospores through mitosis. So these carpospores are actually 2N, because you always end with what you started with. Those carpospores are flung off into the environment, and then they grow up to be the next adult stage, which is a tetrasporophyte. So since the tetrasporophyte grew from these carpospores, this tetrasporophyte is also 2N. Now this tetrasporophyte is going to make tetraspores through meiosis. So if you remember meiosis, we are now going to be in haploid from our diploid cells. So our tetraspores are haploid, even though the tetrasporophyte is diploid. And those tetraspores are going to be flung off into the environment again, and each individual spore is going to grow up to be a gametophyte. Since the tetraspores are 1N, so is that gametophyte will also be 1N. And there's your triphasic life cycle. So that's pretty rare, but it is considered a type of alternation of generation. All right, the next phylum we're going to look at is Chlorophyta, which is your green algae. All right, so green algae are the ones that gave rise to land plants. And one characteristic that they have in common with land plants and not the other organisms that we're talking about today is that their accessory pigment is chlorophyll B, or they have a lot of chlorophyll B as their accessory pigment. And so do plants. Plants also have chlorophyll B as their accessory pigment. So this is another piece of evidence that these green algae are closely related to plants. All right, so the two examples we're going to talk about for green algae are Spirogyra and Volvox. So both of these are colonial species, meaning that they can survive as a single cell, but they do better in a colony. They are not multicellular. So each one of these cells will do just fine on their own. With Spirogyra, it grows more in a filament, meaning long chains. So this is one cell here. You could see the cutoff of the cell wall here and here. While Volvox will grow in a colony that's more of a sphere shape, and here are the individual cells. Okay, they have very different reproductive habits. Spirogyra undergoes something called conjugation, which you will read about in the module. And what conjugation is that you'll have two haploid Spirogyra cells right next to each other. And looking at them, you might not be able to tell which one's male and female. And going back to the types of sexual reproduction we talked about, if you can't tell the male gamete from the female gamete, then that is a form of isogamy. So conjugation is a type of isogamy. However, instead of the gametes swimming around or gametes swimming around to find each other, these are just growing next to each other. And how they're going to transfer DNA is that they're going to grow something called a conjugation tube. And this is the only time you'll be able to tell the male from female. You'll see the male grow a conjugation tube to the female, and then the male is going to squirt all of its cellular content, DNA included, through this conjugation tube and into the female. So now we had this haploid cell and this haploid cell. The male is squirting its cellular content into the female. Now we have a diploid zygote after this is complete. That's different than Volvox. Volvox actually has oogamy, meaning they have a stationary egg, which is a little larger, and a flagellated sperm that is going to swim and try to find that egg. So this is showing you, even under the same phylum, we could have different types of sexual reproductive strategies. All right, one last thing we should talk about is the asexual reproduction. Spirogyra is pretty straightforward. Asexual reproduction happens when each one of these cells undergoes mitosis and adds to the filament. And with Volvox, we can have a special type of asexual reproduction where one of these vegetative cells will migrate towards the center of the colony and start dividing and creating a whole new colony inside this parent colony. And you can see a couple of those right here. These are daughter colonies. So these are clones of this parent colony, just very condensed. And once these daughter colonies grow big enough, this is going to open up and release those daughter colonies into the water. All right, next we're talking about the kingdom Plantae, which falls under the lineage Plantae. All right, just like we saw in green algae, the kingdom Plantae has chlorophyll B as their secondary pigment. Now, one of the major differences plants have that other algae do not is that they have differentiated tissue. And by differentiated tissue, we mean they have actual tissue that do different jobs for the plant. With the other algae, even though they look like they have different structures, every cell is capable of doing what every other cell is capable of doing. Here, we have this differentiation, though. So we have tissue that can have one function, so certain cells that do one thing, while somewhere else on the same plant, we can have a group of cells that are doing a different job for that plant. Now, one of the major differences that you should pay attention to as we go through this is how plants have adapted to live on land. These adaptations are going to distinguish plants from all the other protists under the lineage Plantae. Okay, so the first group of plants we're going to talk about are non-tracheophytes. Tracheo means tube in Latin, so we're really saying they don't have vascular tissue by calling them non-tracheophytes. And this is a more ancient lineage. So the first plants that ever first started growing out of the ocean were non-tracheophytes. They did not have vascular tissue. So this group split off more distantly in evolutionary time. All right, so non-tracheophytes don't have vascular tissue. They also don't have true roots, stems, and leaves. So what does this mean exactly? Well, roots, stems, and leaves are organs. They're plant organs. And the definition of an organ is you need two or more tissues working together to achieve something. Well, they do have something that's like a root. If you took their type of root and cut it in half, you're not going to see tissue layers. You're only going to see one type of tissue there. Same thing with the stems and same thing with the leaves. So because there's only one tissue for their root-like structure, we don't call them roots. However, when we get into more complex plants and you cut a root in half, you'll see multiple tissue layers, tissues working together to create that root organ. Okay, so since they're not true roots, they don't really have stems, but they're not true leaves either. We have different names for them. Instead of roots, we call them rhizoids. A rhizoid is a root-like structure that only has one tissue. Same thing with the leaves. We actually call them thallus. A thallus is a leaf-like structure that only has one tissue. It doesn't have multiple layers, so it's not a true organ. While these have differentiated tissues for their different structures, each one of those structures don't have multiple tissue layers, so they're not true organs. All right, so since we're talking about plants here, they undergo alternation of generations. They have a gametophyte stage and they have that sporophyte stage. With the non-tracheophytes, it is the gametophyte stage that is dominant. They spend most of their time in a 1N haploid stage and then have a very brief sporophyte stage. So the mature plant that you are seeing here are 1N or haploid cells that are making up this plant. Their sporophyte stage is very reduced and brief in their alternation of generations. All right, the next group under Plantae we're going to cover is the seedless tracheophytes. So tracheophyte meaning that they evolved vascular tissue, all right, but they have not evolved seeds yet. So the seedless tracheophytes broke off a little bit before seeds evolved. So this is a little bit more ancient lineage. So first we had the non-tracheophytes, which is the most ancient of the plant lineages we're talking about here. Next we have the tracheophytes that are seedless. Okay, with the non-tracheophytes and the seedless tracheophytes, they both relied on flagellated sperm for fertilization, meaning they're limited to only being able to fertilize during wet seasons. So they have to live in very moist environments and allow their sperm to swim to an egg to find an egg. However, with the seedless tracheophytes, we did evolve vascular tissue now. And by vascular tissue, we mean xylem and phloem. And that gave a huge advantage to these plants. Since they have these tubes that are running through their body, they can actually grow up taller now. That is a limitation if you did not have vascular tissue, because water can only go so far unless you have a structure or an organ that's going to allow that water to move higher up the plant. So this vascular tissue is giving them the ability to grow a little taller now since they have these tubes throughout the body. So the last clade of the kingdom Plantae we're going to talk about is the seeded tracheophytes. And there's two major types of seeded tracheophytes. There's gymnosperms and then there's angiosperms. So both of these are going to make seeds. However, how the seeds are housed is going to be different between the two of them. Gymno means naked in Latin. So by gymnosperm, we mean naked seed versus angio, which means vessel in Latin. So angiosperm means vesseled seed. So what vessel are we talking about with the angiosperms? Well, we're talking about fruit. So in angiosperms, a fruit is going to be formed around the seed or seeds. Well, in gymnosperms, there is no fruit. It's just the seed itself that is released into the environment. With the gymnosperms, they also have unique pollination. They use wind for pollination. So pollen is what's going to carry the sperm cell. And with gymnosperm, that pollen is picked up by wind, brought to wherever the egg is of another plant, and then that sperm cell is going to be able to release its DNA into the egg. But that pollen is transported by wind. Unlike angiosperms, angiosperms use pollinators, meaning insects and animals. So an animal or insect is going to go and pick up pollen and then transport it to another plant where that pollen is released, and that sperm cell in the pollen can release its DNA towards the egg. So since angiosperms use pollinators, they need to be able to attract these pollinators somehow. And they're able to do that by creating flowers. So that's one of the major reasons why flowers are so pretty and smell nice. It's because they're trying to attract animals and insects towards them to pick up this pollen. And then finally, angiosperms undergo a special type of fertilization called double fertilization. And in general, double fertilization occurs when the sperm fertilizes the egg when a pollen lands on a new flower. And then a duplicate of the sperm's DNA is also going to fertilize the nutritive tissue that is going to feed that zygote. Okay, so just looking at these two again, so we're looking at gymnosperms and angiosperms. When we're talking about gymnosperms, there's a bunch of different types, but a pine tree is a good example of one. In a pine cone is where you're going to find that seed. So this seed is not housed in a fruit. Well, in angiosperms, you will have a fruit that houses the seeds in some way. So you could either have one seed or multiple seeds in this fruit, but they're going to be housed inside a fruit in some way. All right, that's it for the lineage Plantae. Now we're going to go over the lineage Strongymanipula. And remember, with our synapomorphies with the lineages, Strongymanipula has hairy flagella. That is their synapomorphy that really separates this lineage out from the other ones. So the first phylum we're going to cover for Strongymanipula is Bacillariophyta, which are your diatoms. Now, diatoms are autotrophic organisms that are aquatic, and they are unicellular and can create blooms. And something unique about diatoms is that their cell walls are made out of silica. And silica is one of those chemicals that are also found in glass. So their cell wall is actually transparent, much like glass is. And this is very beneficial to these diatoms. Because they're photosynthetic, they need to be able to get light to the cell. But by having a glass cell wall, they get the benefit of having a cell wall. They get the protection of having a cell wall, but still are able to have light past that cell wall and make it to their chloroplasts. All right, these diatoms are also used commercially. So you might have heard of diatomaceous earth, which is really a powder that is made up of all these little tiny organisms, or the cell wall of these organisms. And these cell walls are actually really abrasive. So a lot of sandpaper or other abrasive tools that we use will be made up of these organisms. All right, the next lineage we're going to talk about is Mycota, which is your water molds. So in the beginning, people thought they're related to fungi, which is where the word mold comes from. But once we were getting pretty good at looking at molecular data, we realized that they're actually more closely related to the rest of the Strongymanipulates and not to fungi themselves. However, the reason we talk about this phylum in this class is because a lot of them are parasitic and can cause a lot of problems in different environments. You may have heard of the potato famine in Ireland. Well, that was actually caused by a type of Mycot. Another type of parasitic Mycot is fish ick. So fish ick is a disease that occurs in a lot of different fish that is highly contagious, where these organisms will grow off the fish And the fish looks very hairy, but that's because these organisms are growing out the fish, digesting the fish from the outside in. So if you ever plan on a career in ecology, you might run into these organisms at some point. All right, one unique characteristic they have is they have flagellated zoospores, meaning they have spores that move around. That's all that means by zoospores. And they move around using a flagella. All right, but why aren't these considered sperm? Well, because they're not gametes. These spores are not going to fertilize each other. Spores do not fertilize. They're going to swim off into the environment and grow up to be a whole nother adult somewhere else. And then the last group that we're going to cover is the phaeophytas, which are your brown algae. Now, most phaeophytes are multicellular, and the reason they look brown is because of the secondary pigment that they use. So unlike the other organisms, the dominant secondary pigment that phaeophytes use is fucoxanthin, which is more of a brown pigment. However, these are very plant-like, even though they're not under the lineage Plantae. They have these structures that look like leaves, and they have structures that look like stems, but they're not actually true plants. And if you remember, with plants, they had differentiated tissues. Here, you don't have differentiated tissues. This leaf-like structure, all the cells found here can do anything that the cells found in the stem can do. There's no differentiation between these cells. They all can do the same jobs. Their structures just look different. So 2 examples of brown algae are bulk kelp, which you're looking at in this picture, which is found in California and can grow 1.5 inches a day, or sargassum here in Florida. So if you've ever been to the beach, you've probably seen sargassum on the beach. And that's because we're right next to the Sargasso Sea, which is a dense forest of sargassum that's growing right off of our coastline. Now, another reason we really bring up this phylum is because both bull kelp in California and sargassum here in the Atlantic Ocean are habitat producers. All right, so they have created their own habitats here. So they are important species ecologically. Okay, so this was the last lineage we're going to cover in this PowerPoint, and hopefully this helps clarify the relationships between these organisms and what structures they have that are in common and what structures that are unique.